Welcome to this video on CRISPR and Cas9 system. Today we're going to look where this technology originated from, how it works, and finally my opinion on why I think it is a revolutionary technology rather than an evolutionary one. So first, let's take a look at where this system originated from. So this system is found naturally in bacteria as a self-defense mechanism designed to fight against bacteriophages. Bacteriophages infect a cell by attaching itself to the bacterial cell wall and then injecting its DNA material into the host cell. Remember how we learned about B and T cells in the human body and how they can learn to recognize proteins to destroy pathogens? Well, bacteria, they do have a similar system where they learn how to protect themselves from future infections. Mojica first recognized that clustered, regularly interspersed short palindromic repeats, otherwise known as CRISPR, matched snippets from the genome of bacteriophages. He hypothesized that CRISPR is an adaptive immune system. 2 years later, Barangu et al. Confirmed that CRISPR is indeed an adaptive immune system and that it enables a bacterium to take up genetic material of phage DNA into its own DNA to fight off the same bacteriophage next time it attacks. So let's talk more about what this actually means. The CRISPR-Cas defense mechanism can be broken down into 3 stages: adaptation, expression, and interference. When bacterial cells do survive a viral infection, they chop up the viral genome and implant into its own genome. And some of the DNA of the virus that try to attack the bacterium is found between short DNA repeats of CRISPRs called spacers, and during adaptation, these new spacers are inserted into the CRISPR locus. Then during expression, the Cas genes are expressed and CRISPR is transcribed into CRISPR RNA. Now, I just introduced some new words in there, so you might be a little bit confused, but let's take a step back to see what this actually means. So we talked about how CRISPRs are stretches of DNA sequences found in the bacterial genome. Near this genome, Cas genes are also there, and they code for a protein necessary for the CRISPR system. CRISPR RNAs and tracer RNAs produced from the viral infection bind to nuclease called Cas9 and form complexes. The last stage is interference. Here the target DNA sequence is recognized and destroyed. When CRISPR RNA and tracer RNAs bind to the Cas protein and form complexes that cut the DNA of the invading virus, it is able to protect the bacteria from infection. Even after they discovered the CRISPR-Cas defense system in bacteria, there were many questions that were still remain unanswered. For example, scientists were still wondering how the small RNAs and Cas work together to detect and cut the viral DNA. In 2012, Janek and al. Made an incredible discovery. They discovered in more detail how these two things actually work together. They were testing whether Cas9 would be programmed to cleave DNA in vivo, so they dissected how Cas9 and the two RNAs cut the DNA. And what they found out was that the tracer RNA and CRISPR RNA actually paired up to recruit the Cas9 protein before directing it to bind to the target DNA via the complementary base pairing between the target DNA and CRISPR. They found that once at the proper site, Cas9 actually cleaves both DNA strands, and they noticed that this specific location is determined by the CRISPR RNA molecule. And this got them to thinking, what if an RNA molecule could be engineered to mimic the structure of the CRISPR RNA and tracer RNA to guide the Cas9 to cut the DNA at a specific point they wanted? So what they did was they designed an RNA molecule that connected CRISPR RNA and tracer RNA together through this hairpin-like structure. And this engineered RNA actually targeted specific DNA encoding green fluorescent protein. And so they added this engineered RNA molecule along with the GFP protein, along with the Cas9 protein to see if the Cas9 protein would actually cut up the GFP DNA at specific sequences. Saw that fragments were actually there from the cut of the Cas9 protein. And this was a huge discovery because it opened doors for scientists to engineer one RNA sequence, add Cas9, and cut DNA wherever they wanted to. Now, cutting the DNA may not seem like a huge discovery, but it is pretty neat because when the cell tries to repair the broken DNA sequence, it often causes a small insertion or deletion that changes the entire function of DNA. And using this concept, scientists can add or remove specific DNA sequences wherever they want to. Some may argue that CRISPR-Cas9 system is evolutionary in a sense that preceding technologies have been building up to this new one. However, I strongly believe that CRISPR-Cas9 system is truly a revolutionary technology with the power to shift the genetics world greatly. Ever since the discovery in 2012, scientists have been publishing reports at an incredible speed. And just to give you a sense of the craze, Ramana et al. In 2015 found that CRISPR and Cas9 system efficiently suppresses hepatitis B virus. But just 2 weeks before that, Esther Lau published a paper on Nature Review Genetics about how this new technology also defies Mendel's law of inheritance. And this is only the tip of the iceberg. But before I begin, let's first define the word revolutionary. The Oxford Dictionary defines revolutionary as involving or causing a complete or dramatic change. To prove that there was a dramatic change, let's take a look at other gene editing technologies that existed before CRISPR. The two main gene editing predecessors were zinc finger nucleases and TALENs, which stands for transcription activator-like effector nuclease. So both of these were restriction enzymes, and although these technologies were both able to target DNA as well, they were nowhere comparable to CRISPR. To begin with, CRISPR is extremely easy to use, but for zinc finger proteins and TALENs, it is necessary to have a custom protein for each DNA target, whereas CRISPR only requires an RNA programming. Simplicity in target design is a huge advantage to begin with, but on top of that, CRISPR is much more efficient. CRISPR has the ability to make in vitro changes, which means that scientists can directly inject RNA encodings into developing embryos. This would eliminate the long and expensive process of transfecting and selecting certain cells required to create targeted mutant using the previous gene editing techniques. Lastly, one advantage that CRISPR has over zinc finger and TALENs is that it allows for mutations to be introduced in multiple genes at the same time. And this allows for scientists to not only save time and money, but also develop more complex disease models. So, when comparing CRISPR to its gene editing predecessors, I believe that CRISPR wasn't just evolutionary, where it slowly developed over time from previous technology, but rather there is a clear distinctive advantage CRISPR has over the rest, which makes its discovery a dramatic change, thus, a revolutionary change. Finally, I think one of the most revolutionizing aspects of CRISPR is the endless possibility of changes that can undergo through this one technology. Let's go back to the discovery of suppressing hepatitis B through CRISPR-Cas9. So hepatitis B is a deadly, prevalent, and seldomly cured infection caused by a viral DNA in cells. But with the newly developed CRISPR-Cas9 system, Ramana et al. Designed a specific guide DNA that cleaves the viral DNA and reduces not only the viral DNA itself, but also other parameters of viral gene expression and replication. Because CRISPR is such a revolutionizing technology, it has been a hot topic to debate about. For example, China reported in April this year that they would be using CRISPR technique to change the genome of human embryo to study HIV resistance. This started off a worldwide debate about the ethics, and then a couple of months later, UK officials approved an embryo editing study to better understand early human development. CRISPR-Cas9 is a promising technology that could lead to major changes in various fields. However, because it is such a powerful technology, it is important to analyze the risks vigorously while questioning the ethics behind it. And because of the endless possibilities, I am uncertain as to where this new technology will take us. But what I am sure of is that this modern technology is truly a game changer that will start a new revolution.